Hi, everyone. Okay. I hear okay, now it's fine. Perfect. Yeah. Okay. Hi, everyone. It's really a great pleasure to be here today. And I just wanted to start my talk with thanking the organizers because it's the second time I'm here and had the pleasure of already give a short talk in 2019. And it's really a great community and a wonderful event. Most of you have no idea who I am. I'm Philippe Schwaller from Switzerland. I did an undergrad in material science, mostly computational material science at EPFL. Then after short experimental work at EMPA, I joined IBM Research, and that's where I spent 5 years working on machine learning for chemical reactions and synthesis, doing a second MPhil in Cambridge and a PhD at the University of Bern, at the same time. And since last year, I'm back at EPFL, and it's super exciting because I'm building up a super strong team to work at the intersection between machine learning and chemistry. And it grew a bit quicker than I had expected, and I have really wonderful team members and a lot of diverse people. Do you also hear the echo or no? Okay, so I will just continue like that. So in chemistry and material science, we have a huge chemical space to explore, and there are different strategies, and machine learning is definitely one of them, how we can accelerate the discovery cycle. One of the first questions that we can ask is, what material or molecule do we want to make? And then the end goal should be experimental validation. But this is not always achieved in the in silico studies because the make part is actually super hard to do. And in my past research, I focus on accelerating the make part of the molecular design cycle using machine learning. If we take a typical literature reaction as an example, we can imagine different tasks that we can tackle with machine learning. For example, we can predict reactions, or we can also predict how to make a molecule. And this is called retrosynthesis or multi-step synthesis planning. Relevant to this talk is also maybe the yield prediction task, where we can use machine learning models as surrogate models for Bayesian optimization loop. And yeah, if we go to synthesis planning, one strategy that was introduced in, or got the Nobel Prize in 1990 is retrosynthesis. And in retrosynthesis, what we want is to go from a target molecule to the commercially available known building blocks. And we want to predict this route step by step backwards until we know the complete route. And at IBM Research, I worked on that a little bit, and using the molecular transformer I had presented last time, we basically had a model that could go from target molecules, predict back precursor molecules, and another forward prediction model that would basically score the different paths, and iteratively we can build up multi-step synthesis plans for diverse molecules. Our vision back then was RoboRxN, and imagine you're in the pandemic. You do the creative work of designing a molecule that could be bioactive. Then you connect to the cloud, where you get access to a synthesis planning tool. Not only that, you can also convert every step in your synthesis planning tool into a procedure with standardized actions and also reaction conditions. And if you have access to a synthesis platform, then you could execute those standardized synthesis actions on this cloud-connected robotic platform. Or you can also just use those predict these recipes and execute them by yourself in the lab. Here, let me just highlight Teodoro Leino and the RoboRxN team. While I was working mostly on the machine learning part, I was really far, far away from the hardware and the whole engineering work behind this project. Here is just to show you that this prototype in a ChemSpeed robot actually exists in Zurich, and it executes predicted synthesis for a photoacid generator. But today's talk is not really about synthesis planning, but it's about the hard work that starts after you have the synthesis plan. There are also tons of other AI-driven synthesis planning tools, and here I'm just highlighting one of the synthesis routes to a complex natural product that was published in this work by Kubowski et al. And if you look at all those single steps, you have solvents, catalysts, concentrations, time, temperature, pressure, a lot of different reaction conditions and variables that currently are still tuned by the human chemists, and they require a lot of intuition. And this is also true for any material synthesis, I would say. And how do we optimize such chemical reactions? Yeah, as I said, traditionally it's like expert intuition. Then you can also use grid search or optimize one parameter at a time. You can use random or quasi-random search or adaptive experimentation. And here, just to show you a bit the limitations of random search and grid search, if you look on the right, it's not super visible, but if you recognize those blobs, the red circles, you see how it suffers even in 2D and 2-dimension of the sparsity of space, and it gets worse and worse if you increase the dimensions. And you see how the quasi-random methods better cover the whole space. So if you have to initialize your search space, it's typically better to use quasi-random method like Sobol or Latin hypercube. How can we do better? Bayesian optimization, where you initialize your run with prior knowledge or initial data, then you train your model, which you then provide the prediction of the mean and uncertainty to your acquisition function to select the next points and then obtain your results. And you run this loop until you run out of budget or you converge. You can show how you can explore the design space you've defined at the beginning, and maybe also see some trends if in your design space. As a surrogate models, the function that learns to map your parameters to your objective, and we've seen that in the workshop very in deep details. You can use Gaussian processors, tree-based models, and many more, as long as it predicts the mean and the variance for the unexplored points. Then you have the acquisition function, which basically gives you the trade-off between the exploration and exploitation and selects the next points. And you could go on one extreme full greedy, where you just look at what is the highest predicted mean. Then you have acquisition functions like upper confidence bound, where you basically have this trade-off here in UCB with this beta value that is put on the variance, and you could just go pure exploration on the other end of the spectrum. In chemical science and chemical reaction, there are lots of previous work, and here I'm just highlighting a few from the Mather lab, where they looked specifically at continuous parameters like temperature and residence time and catalyst loading, or other works like Griffin for categorical parameters or multi-objectives in Chimera. Yesterday, we also learned about BOS and how it was applied to lignin extraction, which is also, which was super interesting to see. I think no talk on Bayesian optimization for chemical reactions would be complete without highlighting Abigail Doyle's work, which really made it like very popular with this Nature publication in 2021. And recently they updated the work with a web app that does multi-objective Bayesian optimization. But when we moved into this field, and my lab is not very old, that's why there are only like some preliminary results, we kind of wanted to decrease the entry barrier for flexible chemistry-specific Bayesian optimization. And we teamed up with Ryan that you see here that led this Gauche library for Gaussian processes in chemistry work. And what we aimed for is really to have chemistry-specific Gaussian processes for different entities we look at in chemistry, like proteins, molecules, but also chemical reactions, and handle diverse representations of them, and really have this chemistry-specific Gaussian process library. If you look at Gauche, currently it's built on top of PyTorch, and there are lots of fingerprint kernels that work well with molecular fingerprints, like the Tanimoto kernel is very popular, and you have string kernels that could work on top of SMILES, and also graph kernels that are built for molecular graphs. Let's go, after highlighting Gauche, let's go back to chemical reactions and look at this Buchwald to Hardwick reactions that you see on the top that was published by the Doyle Group in 2018. And there, in the high throughput experimentation study they did, they explored different parameters, like they changed the aryl halides, the additive, the palladium catalyst, the bases, and so on. And how can we now represent this chemical reaction for Bayesian optimization? A very popular choice when you are in such a very constrained space is just one-hot encoding. Here in one-hot encoding, if you look at base 1 and base 2, just as an example, if base 1 is present in the reaction, you would just one-hot encode with 1, and all the bases that are not present would get a 0. And like that, you would get in this case, a 44-dimensional vector with all the different components. What the Doyle group is pushing a lot are QM descriptors. They can be local, like atom-wise, or also global on the different molecules. And recently, we've introduced some reaction fingerprint that basically take the complete chemical reaction as an input and directly convert it to a fixed-sized vector. And in this case, as I said, the one-hot dimension would be 44. Quick word on reaction fingerprint and DFT. The reaction fingerprint is a data-driven reaction fingerprint that was learned on a huge database on chemical reactions. And this basically, the encoder model is a BERT model, which is a transformer model and very similar to what you know maybe from ChatGPT. Not exactly the same, but a very similar architecture. And it could technically be fine-tuned on your problem of interest. On the other side, we also worked a lot with DFP, which is a heuristics fingerprint, where we basically subtract shingles from the reactants from the product, and after hashing the remaining shingles and using modular to get it to the targeted size, we get a binary fingerprint back that describes this reaction. Bojana, my student, worked on with gauche to use those different reaction fingerprints, and made some experiments on the Buchwald-Hardwick dataset I was just presenting. And the task here is to optimize chemical reaction yield, like what you get, as the output you get out of your reaction. And it seems that BIO works, but as also observed by other groups, in this case, one-hot encoding, like this uninformed representation of the different components in the reaction, works surprisingly well if you look at this green curve here. And this is certainly because of a constrained design space. This is just to highlight another study that did exactly the same observation. So what we thought is, how can we go beyond one-hot encodings and basically come up with a situation where one-hot encoding doesn't work anymore? And here we looked at a recent publication by the Macmillan group, where they basically screened additives to improve the yields of a nickel photoredox catalytic reaction. And all the other reaction variables besides this additive were kept constant. And if you look, it's kind of a challenging data set because there are only a few or additives that really improve the yield. And if you think, if you have just one class where you change, when you change the components, and you use one-hot encoding, then you just get those 720 different ones or zeros, but it performs as good as random search because you have nothing to learn from. So yeah, we went and set up this experiment, Bayesian optimization versus random, and Bojana went through several different GPs, representations from molecular fingerprints, QM descriptors, learned molecular fingerprints, the reaction fingerprints I was just presenting, and different initializations. And yeah, in the end, she did the model selection on plate 1, and where we had 3 other reactions to basically validate what we had observed. All you see here is with 10 initialization samples and averaged over 20 seeds. And the Bayesian optimization approach using the DRFP seemed to work well on all the 3 plates. What we observed is that none of the single molecule representation worked for any of those, and it's not shown here because in the preprint, when we submitted this to a workshop, basically the reviewer was like kind of a bit annoyed about like the performance, and we removed it and thought we're gonna check it again before like re-putting it out there. Yeah. What worked best for us was a heteroscedastic GP with a Tanimoto kernel, the DFP, UCB with a beta value of 0.1, and a distance-based initialization scheme. If we look at the points it selected during the optimization, we see a clearly higher values for the BO. And also, if we look at the amount or the percentage of runs where the top one was chosen after 100 BO iterations in this data set containing 720 data points, it performs much better than random on any plate we analyzed. If you want to interact with Bojana, she has just put a tweet online yesterday, so you can also go and follow her on Twitter and ask questions on this work on Twitter. What we've realized when working on chemical reaction optimization is that there were quite a few challenges. Here, what I've shown you so far is really on data set that came from high throughput experimentation, and where we basically knew all the things that, all the information. If you then set up a prospective study with an experimental group, as we are doing now with Jeff Gore from my group and the WHO group, it's kind of different. There usually you tackle something, a very challenging reaction, and sometimes even multi-objective. And there's a huge design space, because when you go to an experimentalist and you say, try to map out all the possibilities that could be done potentially, we ended up, even after constraints, with 32 million reactions. And with so many reactions, you have other challenges that come up. For example, if you want to do batch acquisition, suddenly, like doing something like the Kriging believer, where you basically select one point, then you get the prediction by your surrogate model, you add it to your data set, you retrain the surrogate model, and you select the next point again. It becomes suddenly kind of expensive, even in computational time, if your model training and the predictions on the 32 million reactions is computationally expensive. And then always the question, how do you evaluate such a run? Because in the situations where you have all the data, you can easily run 20 seeds and see that on average you're better than your baseline. But in reality, you cannot have 20 chemists or running reactions in independent fashion to really compare with a baseline. And I just wanted to highlight that this is not particularity of this specific reaction, but if you look at this recent Merck paper, basically they also mapped for this palladium catalyzed cross-coupling CN bond formation reaction. They also mapped out 15 billion possible reactions. How I am with the time? Okay, I see. Perfect. That said, I just wanted to highlight another work from Oliver Schilter, a PhD student together with IBM Research. And his task is now not to optimize chemical reactions directly, but to design some catalysts that could lead to a better chemical reactions. And he was focusing on Suzuki cross-coupling reactions, using kind of a variational encoder scheme as inspired by this fantastic publication by Gomez-Bombarelli et al. On automatic chemical design. And what he observed, and what was kind of, I think, the most interesting part in his work, is that when he had only this variational autoencoder on top, then the latent space that he was constructing in the middle was kind of just everything was overlapping and there was no structure, probably as expected. But as soon as he had also a predictor for the oxidative addition energy, which is the rate limiting step in this catalytic cycle, as soon as he activated this predictor, we could really observe supernice structure within the latent space and basically sample catalysts from the desired region. I will just end with a few slides with some recommended resources. There's this recent review from my group, which is kind of short, on Bayesian optimization for chemical reactions. But at the same time, I also want to highlight that work that is much more complete by the group of Alexei Lapkin, who also was published last week. And if you are interested in machine intelligence for chemical reactions in general, there's this review we had written last year. I think like one of the most inspiring resources that I've seen in the last 6 months is also this very nicely done optimization tutorial playlist by Sterling Bert, who is a PhD with Taylor Sparks. And in addition to all the amazing tutorials you got already during this workshop, if you're interested in having more, this is definitely somewhere where you could have a look at. A few take-home messages. I think BIO is very promising for reaction optimization or synthesis optimization in material science in general, but initialization, like the definition of the design space that you have, and also the representation of your molecules, your reactions, your materials, your synthesis is really key. And that's what makes the difference in the end. If you have this sorted, I guess that you can almost take any acquisition function or surrogate model and get to good results. I think one of the open questions is really how to jumpstart the search by using maybe human prior knowledge and knowledge from the literature. Another one is kind of where to place the human in this complete loop. Can we like have some synergies that benefit the optimization? And really this evaluation part, at least in prospective studies, I haven't seen a great solution for that for now. And if you have one, I'm happy to talk about that. With this, I just want to thank all my amazing collaborators and my fantastic group at EPFL and you for your attention. And if you want to know more about my group, just follow us on Twitter and check out the webpage. Thank you very much.
Okay, thanks a lot, Philip. Are there questions, remote or in person? We have plenty of time. Thanks for staying well in time. Army has the first.
Is this okay already? Yes, good. Yeah. So,
is it on?
Can you hear me? Yes. I just didn't speak closely enough. Yeah. So, this might be a bit silly question from somebody who has a physics background. So, you mentioned in one of the last slides that you had some like input variable that you added, and after that the VAE started performing, or the latent space started performing better. So like what was the like specialty in that?
Okay,
or did I get it wrong?
No,
completely?
no, it's not an input variable. Let me just, I think it's here. It's basically we had DFT calculated basically oxidative energies for some of the catalysts in the training set. And instead of just doing an auto encoding from like catalyst representation to catalyst representation and trying to optimize only for that, we basically also had a predictor module that would, from the latent space, predict the energy directly. This is the part that you have in red underneath, and using this second supervision signal, it basically structured the latent space quite nicely. Yeah. So it's not an input, it's more like an output that we,
Yeah. So different training, or the whole setup is different.
Yeah, this like a second training objective that is added. Yeah.
Thank you.
Thank you.
Okay, hey, so there's a question from Matti in the chat. Do you use training data from DFT calculations at any point in your various workflows?
Okay, here definitely this is based on a DFT study published by the co-member group. We in the work that we have in collaboration with the WHO group, we don't use DFT descriptors, but we have some proxies that are simulated with type binding. Yeah. Then in all other, what I've shown before, we try to have as cheap representations as possible and really be as independent from simulations as possible. So when we have those reaction fingerprints, we typically just get a descriptor out of chemical reaction. And the advantage there is that you could have an additional component. It doesn't change anything to your representation size, to your vector size. Yeah, it's what worked well in the past with DFT descriptors and in those data set, for example, the Buchwald-Hardwick one I was presenting, is when not only like global molecular descriptors were calculated, but also like very specific descriptors for single atoms that were human tagged within like this the specific classes. And I think if we really want to have a general tool for Bayesian optimization, we should really have something where we don't need this human labeling at any point. And yeah.
Gareth Condrit from the University of Cambridge. Thank you very much for the excellent talk on this chemical reaction design. Just wondering whether there would be other considerations that a chemist in the lab would want you to ultimately put in, like the toxicity of the chemicals that come out at each step, the side products that could be formed, and how difficult they would be to filter out. And so it's really curious about how you might be able to put those into this in a more generic framework in the future.
No, it's a very good question. Like I have some other projects where we're working on not really on toxicity for the moment, but on more on like side product and byproducts. And I think this is really crucial information that could more go into the synthesis planning work, because I guess there you want to have this information available to prioritize specific paths over others. Yeah. Thanks a lot for the question.
Hello. Hi, Philip. Thank you. Lovely talk. I have 1.5 questions, if that's all right.
Yeah,
the half one to start with is, can you make use of all the sort of failed reactions or intermediate sample steps and creating sort of more public data sets and things of these reactions?
Okay, yeah, I think the goal of, okay, usually I'm not running the reactions myself. My group is currently purely digital, but I think definitely I would like, those are usually like NCCR catalysis collaborations that I have between my students and another group within the NCCR catalysis, and there's a huge push towards open access data and better and better data publishing. So we'll definitely take the responsibility to like show our experimental partner how to best publish their data together with the publication. Yeah,
that's great to hear. Yeah, thanks. The follow-up question is about, you said how difficult it is to compare these approaches to baselines when you're doing something completely new. Like, at what point do you just stop caring about that and just say, we're going to do some chemistry with this now, and you can just get away with publishing it without having to do like a dummy random search?
Okay, it's a very good question. I guess like we can kind of try to figure out what the best methods are on those like well-established data sets, and they're really compared to baselines. But at some point, if we really want to show the value also in collaboration with experimental partners, I think like the the main thing there will not only be the machine learning approach that we have provided to do this reaction optimization, but it's actually probably the most interesting scientific outcome of such a collaboration will be the reaction that we have actually optimized. And there, I think it's fair not being able to compare with other baselines, because it's been kind of used as a tool to accelerate discovery.
Okay, so there's a follow-up question from Kevin regarding the question from Matti. What is your taking in using of different fidelity, such as different experiment sources or some from experiments and some models?
That's a great question. Yeah, no, this is something I'm thinking about a lot, how to like maybe use reactions that come from the literature or from patents with kind of and mix them with reactions that come from the actual experimental group, and but not weight them the same. I think, yeah, that's definitely an interesting area. What I've observed is that the reactions that come from, the most valuable reactions will always be the ones that come from these specific groups, because there are so many unknown variables, I would say, that that are not reported and that are kind of constant within a group, but then as soon as you compare to other groups, usually everything changes. And yeah.
One question for me as well.
Yeah.
I was curious about something that was in your conclusion slide, but it wasn't in the rest of your presentation. Can you guess what it is?
It was more
Outlook.
not only conclusions,
Yeah,
but also
outlook.
just general thoughts about that I sadly cannot discuss tomorrow at the panel session.
Yeah,
but,
exactly.
Yeah.
That's why I wanted to ask you this today. So what are you thinking about the human? How can the human contribute to this loop? In which form would you sample the human?
There are many ways I can think of sampling the human. No, but yeah, no, I think what we've seen so far is that optimization works best when you have this super constrained space, and this is all human intuition. Those decisions are made by the human, so currently it's still super important, and I haven't seen any great models that basically give you the space you should explore, but I think this could be also an interesting area. And then I guess that there might be some synergies in like having, kind of at every iteration, also including a human, and I cannot disclose too many things, but we have some ideas that we are working on.
I guess what I also want to ask you is, how do you leverage the human intuition and exploit it without encoding the biases that we've long had, and not allowing the data-driven models to find genuinely new solutions?
Yeah, but I think you could potentially have like a balanced set of next experiments that are kind of that are not fully human biased, but also not purely machine learning, acquisition function chosen. I don't know. There are many different ways, and I'm sure there will be some publications going into that direction soon. Another thing that clearly benefits from a human is that often we have a hard time defining the scoring function for your problem at hand, and I guess like maybe some human intuition and could go more into this scoring function. And yeah.
Quick question: do we have our next speaker, Simona? Are you here? Okay, good. If you could come to the front. Okay, so we're at the end of the time slot. So let's thank Philippe again. Maybe I can announce to all